Yes, I think it's working again. Hi, hello everyone. Yes, I can hear myself as well. Hi, hello and welcome. Microbehunter here. Welcome to another Saturday microscopy live stream. I'm doing as every time a little bit of housekeeping, getting everything sorted out here. I hope that the sound works again. So let's move the microphone around here. Yeah, that's the thing I'll be doing today. Today I would like to try something a little bit crazy. It's not quite the thing that I planned to do. A little bit of background I will tell you in a couple of minutes. I see that many of you have already started to join the chat. So I want to, first of all, before I get started, say hello all around the world. And wish you a nice weekend. Right now here in Central Europe, where I am, right now the weather is pretty bad. Just a few days ago it was quite hot still, and now we're experiencing floods. That's also one of the reasons why I'm now wearing my turtleneck shirt. So that is completely beside the topic. Today, I actually planned to do something different, so maybe I'm going to catch up on that in the next couple of live streams. But look, this is actually what I planned to do. Is that I wanted to show you how to extract diatom shells using hydrogen peroxide. Yeah, but it's not quite finished yet, the reaction, because my hydrogen peroxide was not quite strong enough, and it takes more time. And for this reason, I had to, relatively on short notice, change the topic of today's live stream, because actually there is nothing yet that I can show you. So I'm going to leave this a little bit for a later episode, and therefore a little bit a slightly crazy thing today is I would like to do this hot chip challenge, but I'm not going to eat this stuff. I'm going to mix it together with a couple of small worms, microorganisms maybe, and see what happens. So that's a little bit more on the I don't know on the fun side. It's not very not very how do you say, scientific, I understand, but in any case, I want to give it a try, and we're gonna see what happens. And this also gives us the possibility to observe again a few live microscopic animals or microorganisms as well. Okay, so for those of you who are new to this format, every Saturday I am doing a live stream about microscopy. I also publish other videos. Please feel free to post questions in the chat. And for the first hour, I'm not going to answer those questions, but after approximately 1 hour, I will go through those questions and I will try to answer them, even if they're a little bit off topic. Okay, as long as they're kind of microscopy related, it'll be fine. Yeah. I quickly, every now and then, skim through the comments as well. Let's have a look at what we're doing here. I want to give you a little bit of background material first. First of all, recently I saw that there was this machine in the village where I live, and they actually sold this stuff here. And I've heard about this from the internet. And generally, I'm not a big fan of challenges like these, because people actually can become ill. But I could not restrain myself, and I actually bought this here, €8, for a single chip. And some time ago, I already made some video where I used a fresh chili pepper and I mixed it with paramecia. The paramecia didn't like it, but there's actually a slightly more serious aspect to the whole thing as well, because the capsaicin, the substance that is spicy, is actually also antimicrobial. So that is also one of the reasons why in some countries, many countries, they're actually putting spices into the food, because it actually helps preserve the food at hot temperatures. And I would like to test this as well. And I'm going to, this has to be, this is basically where I want to play a little bit teacher. Look, that is the chemical formula here of the substance that's so spicy. Okay, if you don't know chemistry, that's perfectly fine. But when I looked at this formula, I was already a little bit worried, because what really worried me is this, look at this long hydrocarbon chain over here. And this basically means that this substance, that's the spicy substance, is not very water soluble. Okay, so this is long hydrocarbon chain here. I'm kind of worried of if I mix now this spicy chip with water, maybe it's not going to dissolve the substance very well. So we're going to try that out in any case, and then we're going to see how well this works. So generally, if you're not familiar with this hot chip challenge, it's like this, that in here, in this box, there is one single chip which is extremely spicy. And then you're supposed to eat this, and then you're supposed to publish this, I don't know, on social media, and I don't know, but when I basically looked at the box here, there are warnings all over the place. And so this is actually one of the reasons why I'm also not wearing my contact lenses now, but glasses, because in case some of the powder actually goes into my eyes, it's easier to get it out again if I'm wearing glasses. I do not want to get it beneath my contact lenses. Okay, so that's also another reason here. And what I'm going to do now is I'm going to take a small piece of this chip, put it in here into this dish here, mix it with a little bit of water, as I mentioned before, hoping that some of the substance will actually dissolve in the water. And then I'm going to feed it to some of the little critters that I have here. Okay, that's a little bit the idea. Yeah, so generally I don't mind eating spicy food, but I'm not, I don't know, if it hurts too much, then it's also not fine. Yeah, so this is basically how it looks like. Look, my last words. Any last words? I don't know. Yeah. Supposed to have the shape of a coffin, so marketing is all. What do they have here? What is this, some kind of a sticker or tattoo? Here's a sticker. Oh my gosh. Okay. What else do we have here? An instruction manual on how to, I don't know. Yeah. And some advertisements. We don't need this stuff. And some gloves here. Because if you get the powder on your hands, you have to be careful that you don't get it into your eyes, and that's apparently the chip. And I've never opened this before. It's the first time. Okay, interesting. Some statistics, basically what happens over the time. So after 3 minutes, it's supposed to be very spicy. So that's basically the thing here. And I want to take a small part of this and put it in here into this dish. Let me move this a little bit over, maybe, so that it's a little bit better. And I'm going to then put it on under the microscope. I just also wanted to show you because I have already prepared the first victim. And the first victim, I hope, is still somewhere floating around here. The first victim is a flatworm, which I collected from some pond water. And we're going to see how it likes to eat the spice. Okay, so that's the first one that I would like to feed it. And we're going to see what happens. Okay, and honestly, I probably think not a lot is going to happen, but we're going to see. So this is it. Okay, now what? Now I have to open it. You know what? I'm going to open it in a controlled way. I can actually rip it open here as well. And I don't know, should I put on my gloves or not? I've never tried this before. I do have some tissue papers here as well to wipe my fingers. And this is how it looks like. Okay. I think what I'm going to do is I'm just going to break off a small amount first. It doesn't even look that spicy. It doesn't look that spicy, actually. And I'm going to simply put a small piece in here. And you know what I'm going to do now? Of course, you know. It's spicy, but that small amount actually tastes good. No, it's very spicy. Even that small amount is quite spicy. But actually, this was only a very small amount. It's actually, this is, the whole cracker would be definitely too much. But that small amount actually tastes good even. It has an interesting flavor to it. I'm a little bit concerned about something else, though. It also tastes a little bit salty. I'm able to taste the salt because it was not so, I only used a small amount of the spice. And if there's salt in there, I have to be careful because this actually changes the osmotic balance of the organism. So, so that's it. Okay, I'm going to put the rest back here. And now let's give it a try. Okay. Let's give it a try. I'm going to now add a little bit of water here. I still taste. It still tastes a little bit spicy, but I can definitely imagine the whole cracker would be definitely too much. Okay, but this little amount is actually quite fine. Okay, let's give it a try here. It will soak a little bit. I think I added a little too much water though. Yeah, I'll see. Okay, we're gonna see what happens. I can actually see that it starts to dissolve a little bit. It starts to slowly change its color. And yeah, I'm not quite sure whether the water actually will dissolve sufficient. Yeah, otherwise I'll have to add maybe a little bit of this cracker, yeah, and of the chip, and then we'll see what's going to happen. I know this is not a very, how do you say, systematic way of testing this, but yeah, we'll give it a try. And yeah, it starts to slowly swell up a little bit. And then let's put it under the microscope. Okay, I'm going to take, this is a fresh pipette, and maybe I'm just actually going to go a little bit in here. And I'm going to take a little amount here. And I'm going to now change over to the microscope view. Let's have a look where here it is, the flatworm. Yeah, then let me close the condenser a little bit more. There are not so many other microorganisms around. Yeah, you see here, that's the edge of the water drop. It stays in there. The slide that I'm using right now is a concave slide, so that means it has an indentation. And yeah, just see, and this is I'm using now the little nematode worm here. We might also observe this one, and I'm using now my lowest magnification. It's for the 4 times objective. Yeah. So maybe I'm opening it a little bit again. If you want to have some color, then I can add this one over here, this little prism. Let's give it some color. Yeah. So it looks also nice. So, and you see the flatworm is fairly large, and therefore, yeah, it always goes a little bit out of the field of view. So let's see what happens. Okay, really, I have never tried this myself. So just to have a look what I'm doing here on the top. Then you can actually follow along as well. So I'm adding a little bit of this. Yeah, I don't know if this was enough. Here we go. Ah, yeah. You can actually see that here, those little things here are part of the chip. Let's have a look what the nematode worm does. The nematode worm really doesn't care. Okay, and where is the flatworm? And let's see what it does. It could actually be that it actually sees some food, a source of food in those little chip particles, and maybe actually it will feed on it. Yeah. Ah, yeah, you see that there. Ah, also interesting. Do you see this round? I'm going to use an arrow over here. Do you see this here? Where's the arrow? I have to be able to move the arrow now. Just a second. Ah, yeah. Do you see this here? So it looks a little bit like a fat droplet because, as I mentioned, this spice is actually fat soluble. So maybe there are some oils in here as well. And where is it? Here it is. Let's see if it starts to avoid it or whether it actually uses the thing here as a food source. But it seems to kind of seems to stay on the edge here of the water droplet. And could of course also be that it's not necessarily the spiciness. Yeah, see those round fat droplets. It could be any some other substances that are in there as well that yeah, it might respond to. Yeah. It's actually also quite nice to see how the particles in there make the flow of the water visible. I'm a little bit off center because I was, yeah. So I'm also checking a little bit while we're waiting. I'm also checking a little bit. Yeah, look, the flow of the water is not quite well visible. but actually I think it doesn't seem to do it a lot of harm either. Okay. I'm also reading a little bit, and when I'm pausing, then I'm also reading a little bit the comments as well. Green screen. Yes, as a matter of fact, I do have my wall in the background is painted green. It's a green screen, so I can change the background, but I decided not just to leave it green. So, because then it's actually simpler that way. Huh? Yeah, so the flatworm seems to be doing reasonably fine here. So maybe I'm going to, you know what? I'm going to go up with the concentration yet a little bit further. Yeah. So I'm going to pick up a little bit more of this. And let's add some more. Up, here we go. And where is the flatworm? Yeah, the nematode is here. And where is the flatworm? Here it is. So, concerning the substance, this capsaicin, okay, it's like this, that it actually has the possibility to react with the nerve cells in our mouth, which are actually there to detect heat. So it is actually, we're not really tasting the spiciness in that sense. It's not really the taste, but it's actually the sense of heat, because the receptors, the nerve cells, which are actually there to detect heat, can also be stimulated by this chemical substance. And this can, of course, also mean that other animals, which do not have those receptors, or at least receptors that can be stimulated by that substance, they will also, of course, not be able to detect this. And this basically means just because a substance can be detected by us humans does not mean that other animals are able to detect them. Obviously, right? And just because something tastes spicy, it does not mean, well, that's actually a subjective perception, right? That is simply shows that our brain creates this feeling, right, of pain because of the nerve signals that are sent to the brain. Yeah. So, but essentially, it could be quite well possible, and it's very likely that other animals do not sense it because simply they do not have receptors that respond to it. Yeah, and this flatworm, and I was actually hoping to see a little bit is whether there is a certain antimicrobial effect, or flatworms are strictly speaking not microorganisms, but generally because the chemical structure of the molecule is such that it is actually able to disrupt cell membranes. And therefore, single-celled organisms essentially can actually pop open when they're exposed to this spicy substance, because it is able to disrupt the cell membrane. And look over here, this nematode worm really doesn't care. Yeah. So, yeah. So that is so you know what I'm going to do is I'm just going to leave it like this, and I'm going to try it now also on other organisms as well. I'm going to just leave it here, on the side. I just have to make sure that it doesn't dry out. And I'm going to now take another water sample here. Here it is. No, that's not the one I was looking for. Well, maybe it is. There is a gigantic flatworm here. There's a gigantic flatworm in here, but I will try to catch something interesting here, and I don't know if I'm able to do that. But let me try maybe an easier sample. I've got several of those. Okay, so this one over here, this one over here. What about this one over here? There are a whole bunch of snails in here as well. Yeah. But I will try now the next one, this one here. Yep, there's some some water fleas as well. I don't know. They're kind of difficult to catch, so I'm not quite sure whether this is going to be successful or not. Okay, I'll give it a try in any case. Where are they? Did I catch it or not? I don't know. Oh yeah, I don't know. So I think I didn't. They're so rapid and so fast. Let me see if I can get another one. What's this one? Oh, this is, I don't know. This is a huge one. I don't know what that is. This is a huge one. Let's just have a look. Okay. So let's just do it up. I have no idea. Okay. This is, in any case, this seems to be a pretty large one, and let's have a look what it is. Ooh, this looks almost like a mite. Okay. Yeah, crawling around. I have to do the following now. I'm going to now try to take a little bit some of the liquid away, so that it's not able to move quite as much. And then maybe it's going to be a little bit more stationary. So let me take it away again. And I'll just show you again on the desk what I'm doing here. Okay, so I can actually show you here. Maybe you're able to see it. Okay, here it is, this tiny little dot that you see here. Okay, this is now a fresh tip, okay, what I'm using here, and I'm going to simply take some of the water away. Because this will also limit the movement a little bit, and when I add more of the liquid later on, it will also be a little bit more concentrated. So let's have a look again. Okay. Let's have a look again. I need to change the view, scope view. Here we go. Here it is. Okay. Yeah, the rainbow colors are a little bit extreme. Let's put that away again. It's almost a little too fast. Insect or mite, we have to count the legs. Looks very much like a mite, of course, also the body shape. Yeah, and it's got 8 legs. Yeah. So okay, let's try it again. Let's see how it responds. Okay, let me center everything again here. Yep, and here we go. And it also doesn't seem to mind. Still moving around. Dark field, somebody just said. Okay, that's actually an interesting one. Let's see, am I able to get this in dark field as well? Whoop, here we go. Oh, it actually looks, that's dark field. Here we go. Actually looks much nicer because now we're able to see the pigmentation of the exoskeleton a little bit better. Yeah. So in case, I don't know, what do you think? Do you feel now disappointed that there is no reaction, that they don't seem to mind the spiciness? Maybe they're able to detect it, at least there is no significant change in their behavior. Yeah. Are you disappointed because of that, or is this actually something that you expected? Well, I did not try it out, and as a matter of fact, I kind of, almost expected that. Yeah, because there are millions of substances out there, and just because a substance tastes spicy, and that's simply because our nervous system, our receptors are able to detect that. Yeah? But this does not mean that there is an inherent, the substance is inherently aggressive or anything like that. Yeah. Yeah. I'm also seeing that essentially somebody's just commenting that this might be a concentration issue. That is also correct. Yeah. However, I did try some time ago, actually several months ago, where I actually took a fresh chili pepper and I directly put the juice of the chili pepper on Paramecia, on single-celled organisms, and they actually did respond to that. And I can imagine that maybe larger organisms, like for example this mite over here and also the flatworm, because they're made of many cells, it simply takes longer also for the cells or for the organisms to actually absorb the substance, so for it to become effective. So this could also be a reason. But in any case, I think, yeah, it's still interesting and fascinating to observe here. Making a slide, permanent slides of mites and also insects is can be a little bit challenging as well, especially because they're fairly thick. So what you have to do is you have to usually treat those insects and mites. You have to treat in some potassium hydroxide, for example, which will essentially also dissolve the softer parts, the softer organs, and will also preserve the exoskeleton better. Yeah, and then you have to remove all of the water using an alcohol series and so on. So preparation of insects is a little bit elaborate, especially if you want to make permanently lasting slides. Before I forget. I've ordered a few more antique microscope slides as well of cross sections of sea urchin spines. If you remember, those of you who were here who joined me last week, I put some beautiful sea urchin spine slide under the microscope, and I found a few more, and I also purchased them, and they'll be arriving in the next couple of days. And so when they're here, I would like to show them to you as well. Yeah. So I just want to say thank you for your financial contributions, because I'm using those to buy slides which I want to share with you. Yeah. So that is actually a nice specimen again. Some mite, which I have to check my identification book, hoping that it can be found in my identification book. Yeah, but then again, there are so many of them that chances are also pretty good that the organisms that you see under the microscope cannot be found in an identification book. Yeah, so because biodiversity, as I always say, is quite high. Yeah. I just see that there are a couple of questions that I would like to respond to later as well. For example, adding other substances like soap. And indeed, soap generally is a problem because it's an alkaline substance and it also dissolves the cell membranes. Yeah? Something that I also might want to do, but I need to grow again a few more microorganisms like Paramecia, very large ones, which are easy to observe then under the microscope. So this is the one. Let's see how the other one. Where's the worm? Here it is, the flatworm. That's the flatworm now in dark field. Yeah. Doesn't seem to mind at all. And all of those dots in the background are actually, pieces of the cracker, of the chip. Yeah. Doesn't seem to mind at all. Yeah. Doesn't seem to mind at all. So here it is, the other one. Again. Yep. Okay, you know what? I'm gonna move on. I have another worm called Iolosoma, which is also quite a nice one. So what I'll do is I'll simply take those slides away and put them here. And later on, some people are a little worried. Later on, I'll put them back, of course, into the water container. So there's a gigantic snail over here, gigantic flatworm. I don't know if I'm able to catch this guy here. Let me see. Just a second. Okay, let me hope for the best here. Ah, yeah. Okay, I also got something here that I would like to show you. I think many of you have already seen this before. So where is this? Oh yeah, here it is. Huh? Where is this? You of course know this one. This is an ostracod. Yeah? It's a crustacean. You know what? I kind of like the dark field. Yeah. And I think I'm just going to leave it here. I wanted, for those of you a little bit technically interested, what I actually have is a so-called a phase contrast setup, and I also have a DIC. It's a differential interference contrast setup. But for phase contrast, you have a certain filter. And this filter for phase contrast can also be used for dark field. Yeah? And that's basically what we got here. The guy stopped moving. Why did he stop moving? Sometimes they stop moving if there's too little water present. But I mean, you still see a little bit of movement here. Let's add a little more water. Maybe it's kind of sensing that there's too little water. Here we go again. Okay, so now to the chili. Okay, here we go. And where is it? And let's add again a little bit of this. Here we go. Ah, that's interesting. No, here we go again. At least right now, also doesn't seem to mind. It doesn't even seem to avoid it. Maybe. Quickly zipping around. Okay, what I can do, and I don't have I don't have dark field for the next one anymore, so I have to move this away. This one up. So, pop, well, difficult to chase, and out of focus as well. Now I think I'm just gonna stay. I think I'm just gonna. Oh, where is it? Gone. No, I'm going to stay at the lowest magnification. I think that's the easiest one. Yeah, to deal with. Okay, gotta find it again. That's why it's always a good idea not to make the, here it is, not to make the water drop too big. Yeah, so here we go. Yeah, so that's basically the little ostracod who also basically has passed the one chip challenge. Yeah. And yeah, it's yeah, still going. So tardigrade time, somebody says. Unfortunately, I don't have tardigrades here with me, but that's certainly something that I could also try. Okay. So that's an interesting one. What about aphids? I luckily, I mean, I'm in our garden. There were some bushes that were growing some fruits, the so-called the red currant fruits, which are quite nice tasting. And a couple of years ago, 2 or 3 years ago, there were so many aphids on there. And they really damaged the plant. And luckily, we got them away. And what we have done is apparently this also works that if you spray the plants with soap water, apparently the aphids don't like that. And we tried that, and this actually did what I assume did do the trick, and then they disappeared after some time. So yeah, so this one also passed the test, and I'm going to try to find another one now. Okay, so let's take it away. So that's slide number 3. So, and where is the desk view? Here we go again. Okay, you see how the chip actually has soaked up quite a bit of What I have to tell you, what I tried to do before, not using this one over here, but actually used real chili. I took some of the chili and it was very hot chili, and it dissolved it in a little bit of water, and then I tried to taste a little bit of the water, and indeed, it did taste a little bit spicy. So actually, some of the substance does dissolve, but not very efficiently. Now, what you could do is you could add a little bit of alcohol to make it to dissolve it better. But then you've got the problem that the alcohol actually might damage the organisms. Yeah? So that's also something you don't want to do. Yeah? There's a whole bunch of water fleas in here. Just a second, I'm checking where are my little that's the one. Yeah, so that's the one. It contains many iolosomas. Iolosomas are those little cute little worms. So-called the oil droplet worms, and there are, for whatever reason, many of them in here. So this shouldn't be a problem to pick out one of them. They like to be on the side of the... Up here we go. Actually, I think I got two. Okay. And let's see. One. Two. So iolosoma, that's a so-called a segmented worm. So quite different from the flatworm that I showed you before. And the scope view. Oh yeah, again, dark field, here it is. Let me refocus. That's actually a small one. Yeah. The other day I actually found one where you had quite a lot of quite many diatoms in the digestive system. Where's the second one actually? Here it is. Yeah. Here you can also see the food that's in the digestive system. You know what? Now I'm going to actually switch away from this here, and I'm going to now indeed switch over to the other contrasting technique now, at a higher magnification, because here we see more details. Yeah, but it makes it much more difficult, of course, to chase around. And in this sample we can also see that there are some single-celled organisms. Yeah, they're too large for bacteria, but probably ciliates or flagellates or whatever also in the sample. So that's a smaller one again. Yeah. So actually, if we there is no cover glass on here, so that's a little bit easier for me to add the substance. However, with the cover glass, because then the worm is compressed between cover glass and microscope slide, it's also not able to move as quickly, and therefore it's easier to observe. Yeah? So that's basically iolosoma, I'm going to add now again the substance. Mm-hmm. There are a couple. I'm just reading. I see that there are a couple of very interesting questions there. Okay, that I would like to then address. So let's have a look here. Let's remove this here as well. So we're just in regular bright field again. You know what? Why not? Why not try? Okay, I think I kind of like this as well. This dark field looks nice. Okay, so let's try to add again a little bit some of this spicy substance. So 3, 2, 1, and 1 drop. Yeah. Now can see quite clearly now. Yeah. The particles of the chip. Let's see if it turns away. Okay. Those worms like to reproduce by dividing in half as well. So this basically means that a new head starts to grow right in the middle of the body of the worm, and then the two worms separate. It looks quite interesting. Some time ago, I spent many hours just observing those worms as they reproduce that way. Yeah? So you can actually see that over the hours, the head starts to grow more and more, and then it will separate, and then you've got two worms. Okay. Yeah, but look, this here is now a little bit interesting in the sense that on the left side, that's the end of the droplet, right? And it cannot move towards the left because this is where the droplet ends. But as soon as it goes to the right, when it touches the place where the concentration is so high, it actually turns around. So maybe it is indeed able to sense that there is see, it turns around again. Yeah. On the left side, it turns around because it cannot go further anymore. And on the right side, when it touches that, it seems to turn around because it somehow doesn't like whatever is there. So maybe they are indeed, these worms are able to sense it. Up, see, it turns around again. That's again. This is not a a scientific study what I'm doing here, but still, if you observe, then it might give you a little bit of an indicator of which substances the animals are able to detect. Yeah. But actually, this is also good because they're not moving around quite as much as before, and it refuses to go in there. Or is it? No? With time, of course, the concentration gradient starts to become less and less. So this means the substances spread more evenly throughout the droplet. So. Yeah, some of you are researching in the background. That's quite nice. Yep, here we go. Nothing, no. So, if we go in, let's go up a little bit with the magnification. Let's put this away again. Let's refocus. What you're able to do also is you're also able to follow the food as it passes through the digestive system as well. Also something that can be quite nicely seen, especially because the worms are so transparent. Just yesterday, I've actually spent quite some time making some nice videos of these worms with a cover glass on top, so you're actually able to see many more details. And the worm is typical because it has those reddish dots all over the body. Yeah? And these dots apparently are oil globules. But it's a little bit difficult to see now because it's moving around so erratically. Yeah. So, yeah. Also, apparently, not a very big response. Yeah. Okay. So I'm actually I am waiting for a collection of all of the questions, but there are a couple of interesting points here as well. Yeah. So, for example, if insects or tardigrades are able to detect this substance, yeah, and the thing is the following. Why is it that actually the chilies even produce the substance? What's the whole point? Yeah, and these are so-called secondary plant substances, and secondary means that those substances like capsaicin are not essential for the survival of the plant. So there's so-called secondary metabolites. They're not, but they do serve important functions as well. For example, because obviously, if they taste spicy, then this protects the plant from being eaten. Now, this begs a question, of course, okay? Because those chili peppers, contain the seeds, right? And if essentially animals do not eat them because they taste spicy, well, how are the seeds then spread? Yeah? So that's, or maybe they should only be spread by certain animals that are not able to detect the spiciness. Yeah? Because normally fruits, not in all cases, but in many cases, fruits are there to be eaten by animals so that the seeds can be spread. But evidently, in the case of chili peppers, some animals should not eat them. Yeah. So yeah, you see, nothing happens very much, and but actually, because you can see those round oil globules as well in the liquid, it actually means that there must be some of the substance present. Yeah. Yeah. So you know what? As we look at this here, maybe I'm going to, what I could try is just for the I'm also checking. I have 15 minutes, maybe more. Yeah, I'm checking if there are some other organisms in here. There are plenty of worms. Maybe some in the sediment down there. I don't know. Let me see this one over here. There are. This one is. I've never seen so many Iolosoma worms before as in this one. I think they have reproduced quite rapidly. So apparently they have quite favorable conditions here. Yeah, but otherwise I don't see a couple of water fleas here maybe. But they're all the same type of worm. So maybe another ostracod, an occasional one. I can also see that there is a little bit of cloudiness on the bottom. This is actually a sign that there could be bacterial growth happening. I mean, of course, bacterial growth happens. I'm well aware of that. But quite a bit. So I've now taken up also a little bit of the sediment, hoping to be able to find something interesting there as well. You know what, I'm just going to put it directly on the slide next, on the place next to the slide. So let's have a look. Ah, what is this? Ooh, yeah, this is a huge ostracod. Yeah. Yeah, that's a huge ostracod here. Is there anything else? Yeah, lots of smaller microorganisms in the background. So what I could try maybe is the following, because we already checked out the ostracod. I think I'm going to remove it again. And I'm going to add a cover glass here and some of the spice to see how those single-celled microorganisms actually behave. So I'm going to remove this again, and I'm going to take off the ostracod again, if I'm able to see it. So here we go. So let's remove all of the solid material and the larger particles here. So that's number one. Where was this? This was here. It's back again. A little bit more. Okay. Then a cover glass goes on top. And then I'm going to be adding some of this spice from the side of the cover glass, and let's see how this works. So this is number one. Oh, I just see. There is still a lot of solid material in here. Okay, not working because there is the cover glass is not fully on top. Let me push this on the side here. Now this is better. Okay, that's better now. Let's have a look. And then we can go up really much with the magnification. So let's refocus. Here we see, now we see those single-celled organisms. Yeah, there are a whole bunch, a couple, this one is in the center. Let's see some diatoms as well. Let's go up yet further. It's a little too dark, I think. It's a diatom. So, yeah, the further I go up with the magnification, of course, the more difficult it is to focus it. But what we can try is when I apply some of this spice on one edge of the cover glass, it will start to form a concentration gradient. And if they don't like it, then they will actually move away from the concentration and will move to one side of the cover glass. So, and that's actually something that I would like to try. Okay, so let's try for this, we have to make sure that we take a small amount here. And now I'm going to try to apply it to the edge of the cover glass. And it will then be taken up by the capillary action. And let me go down a little bit with the magnification here. So this is here at the top, this is where I applied it, right? And the capillary action is drawn in. We can see that there are lots of these particles now here, and the further we go away, and we see that there are none of them anymore, and here they start. Yeah, and usually what I do is I observe them, what happens to them when some of them start to move back to the higher concentrated area. If they do that in the first place, if they do that at all. Yeah. So the further here, there are more of them here, the further away. Yeah. And here at the top, okay, there's an air bubble in the way. Yeah. And now it could of course be, here's one. It could of course be that they're less there because of the movement of the fluid, that this kind of flushed them away. This can of course be the case, but this one over here is now in the concentrated area. They start to move back and they don't seem to mind. Okay. They don't seem to mind that the paramecia, they didn't like this at all. Yeah, but they started spinning first. Yeah, but they actually seem to be doing fine. So, yeah. So at the beginning there were fewer of them because of the liquid, which kind of pushed them away, and then now that they're able to freely move again, they seem to be going back here, and they don't seem to mind at all. You see again those round globules of oil, which is actually an indicator that indeed there is quite a bit of the substance present. I just read over here that there's a question about salt, and indeed, what you can do is if you apply a little bit of salt water on one edge of the cover glass, this is then paramecia and some other microorganisms will move away from that region to the other edge of the cover glass, and that's one way how you can actually concentrate them. But if you add too much salt, of course, you're going to kill them off. That's also clear. Right? So, okay, this actually shows that a little bit of what I kind of expected, that all of my little critters here seem to pass this one chip challenge. because all of them seem to be doing fine. So what I'm going to do now is I'm going to try to check again the other examples here. Oops, I just see the following. Okay, let me try this one here again. This one actually started to crawl off. The slide started to dry a little bit. What is this? Ah, yeah. Ah, yeah. That's the little mite, which is now having trouble moving because so much water evaporated. Where is it now? Yeah, here it is. Yeah, now it's able to move again. Okay, so that's the mite. It's still doing fine. Then number 1, number 2. So let's see if what this guy is doing here. That is the flatworm from before. Ah. Maybe the flatworm is not doing so fine after all. Or could it be because there is still plenty of liquid here? Interesting. Let me check if it's the amount of liquid that it didn't. Yeah, but there's still. I see that there's still quite a bit of liquid present. And thank you very much. Lots of questions again. Okay. Yeah, I just received your questions. Yeah. Okay, so the flatworm is doesn't seem to like it quite as much anymore. Okay. And the next one, what is this here? I forgot. It was this ostracod. And where did I put my worms? I forgot. The other worms are where? I guess I must have lost them. Here they are. Yeah, they're still fine. Okay. Still moving around happily. Okay, folks, you know what? I think I'm going to move on a little bit, and I'm going to try to go through your questions now. Okay, it's a little bit what I kind of expected to happen. That in most cases there is probably no response, and I have to still be careful because if the spiciness, if the capsaicin is on my fingers, then when you wipe your eyes, then it's not so good. Okay, so but you know what, people? I've got over almost over 1 page, 1.5 pages of questions here. So what I'm going to do is I'm going to carefully move the slides away so that I can put them later on back. Okay, and let's have a look here at some of your questions. Okay. Okay. Shout out. Microbehunter inspired me to start my own microscopy channel. That's actually a general recommendation. If you are visiting other people's channels, please do subscribe to them. Okay. Make sure that essentially that you also like their videos and so on, because this, of course, is very motivating. If you only eat Austrian food, this will hurt. Well, I like to eat a lot of international food. As a matter of fact, I don't even like, I like actually pretty much almost all kinds of food in that sense, including spicy food. Generally, Central European food is considered by many people extremely salty. Okay, the green screen is not set to transparent. That is correct. There's a technical question. Some time ago, I did set it to transparent. And the problem with that was that sometimes also part of my face became transparent because the light was not always very consistent. Okay, so I basically, instead of if you watch some of my older videos, you're going to see that the background was actually dark blue. And then I decided, well, actually, it doesn't really matter what the color the background is, so just leave it now natural as it is. Yeah. Yesterday, I asked a nurse to take 2 blood tests from me. I have a centrifuge. Any suggestions of experiences I could do with it to see in the microscope or experiments? What you can definitely do, and I have made videos of this, is blood is very interesting. What you do is you take a small amount of blood, put it on a microscope slide, cover glass on top, and you try to look for white blood cells. And when you found one, then without moving the slide, just take a video of it for 10 minutes or 15 minutes, and then you speed it up, and then you can actually see the movement of the white blood cell. It's quite interesting. It moves around like an amoeba. But if you just look at it under the microscope, the movement is so slow that it's very difficult to see. But if you then speed it up, then you're actually able to see, that the white blood cells or some of the white blood cells actually move around like amoeba. Yeah, where did you get your the tiny dish? Oh, this one over here. Yeah, this one over here. I think if you are referring to this one, this dish I got from a school supplies company. I'm a teacher, and we have catalogs that basically have school supplies and microscopes, accessories, educational material, and this is where I got this from. They're a little bit difficult to get, but if you Google for block dish it's called, then you're able to get it. However, what you might also try to do is if you don't have those, you can actually get also some small Petri dishes. Here now I've got some cover glasses in here, right? And those Petri dishes actually also work very well. The advantage of those flat dishes is of course that you cannot knock them over and not spill any water. Yeah, but I have to admit that indeed those block dishes are not quite as easy to get. Yeah, but those Petri dishes are, and I would say that they work equally well. How do you remove hard water stains on glass slides? You treat it with vinegar acid. Okay, I have tried to clean those by dipping them in a dishwasher liquid solution, but nothing worked. So generally, the water stains that you have, that's calcium carbonate that deposited, and you can of course use vinegar, but sometimes they also sell. I don't know how it's called in English, but it's basically much more concentrated. It's not just 4 or 5%, but around 20 or 30%. Very strong vinegar, and this also works. Okay, so acid is important. Yeah, and you just let it stand for a little bit, and then you just rinse it, and this usually works. Yeah, but you have to be patient. You have to allow the vinegar, the acid, to do its work. Can you please post a video where you show how to set up crystals and illumination for a microscope? I can't find any videos. I don't know this form of illumination. That's interesting. Maybe it goes under a different name, but thank you for the comment. I will try to research this. Okay. I will research this, what this is, because maybe I do know what it is, but maybe I'm not familiar with the name. Okay, you could use hot pepper flake and water. You want to avoid the salt sugar content. Yes, that's indeed also possible. Okay, so some spicy dried peppers, and you kind of dissolve them, and then you can more easily filter them away. By the way, this is something that I tried before the live stream. I took some hot pepper flakes, added a little bit of water, and I wanted to see if the substance, the capsaicin, is actually able to dissolve out. And the water did taste a little bit spicy. So it does dissolve a little bit in water. How to know the objective power by its color? I can't see the objective power as the OIC is too small. Okay, thank you for the comment. So basically that is the thing here up here, right? This one here, it's like this, that even though it's color coded, sometimes the actual power written on it is not visible because sometimes the magnification is basically printed on the other side of the objective. Yeah. But simply to run you through, I have got let me lower this here a little bit so that I don't crash the objective. I've got a 4 times, 10 times, 20 times, 40 times, 60 times, 100 times oil, right? And another 40 times phase contrast. And then I'm back at 4 times. So that's basically what I've got here. Once per few months, you should invite a few subscribers with microscope camera to get a live session together. That's actually that's a very nice idea and something I also thought about. It would be kind of nice if somehow it were possible to do a live stream together. Okay, I have to figure out how to do this. I don't know how to do this yet. Okay, but it shouldn't be a problem if I don't know. It should be possible if the people live stream it on their own channel, that I kind of include the video also on here in one screen, so it's easier for the viewers to see everything on one screen. I still have to figure out how this is done, but that would indeed be a nice possibility. Yeah, I know it must be possible somehow, but I have to see how this would work. Yeah, I wonder how the worm would react to Tabasco sauce. Yes, the thing is the following: Tabasco sauce contains so much acid and vinegar. So it's very sour. So I'm quite sure that many of those little animals, they wouldn't like it, but not necessarily because of the spice, but maybe because of the other substances like acid, vinegar, and so on that are present. And salt, of course. Could you put hot chili flake on them? Okay, to get them more concentrated. The thing is the following: the chili is probably not more concentrated, because when I just tasted it before, the chili, it's actually much less than, for example, the spiciness of those here. So those crackers here, they are way more spicy, than the chili. Yeah. So I think it's more a question of how can I get it into the water, the chili powder, right? How can you tag people? I cannot tag anyone here. Okay. As a biologist, I'm sure you already knew nothing would happen to them. This is why, you were concerned by the salt. Yes, actually, you were correct. I was actually more worried about other substances than the actual spicy, the capsaicin, right? Because as I explained to you before, it depends, and as many of you have already mentioned, just because you're able to detect something does not automatically mean that it's necessarily just because it hurts you when you have it in your mouth does not necessarily mean that it also has other biological effects, right? And it could be that some substances that don't taste like anything are actually quite harmful. Yeah? So that's the thing. Although will common soap be a problem for microbes? Yes, that's why we actually use soap. And also generally, also mites and insects. Generally, yes, because what soap does is soap is a detergent, and this basically means that it dissolves the cell membranes. Therefore, when you add soap or detergent to microorganisms, then many of them will actually start to pop open and the contents will flow out, and that is the reason why soap actually is able to kill microorganisms. Also, soap is very often basic, alkaline. And this can also be, of course, causes certain chemical reactions to happen in the cells of the microorganisms, and therefore they will actually also die. Larger organisms like mites, small animals, and so on, of course, they also don't like that, obviously, but it might take longer for them to respond because it takes more time for them to take this up. Yeah, vinegar, same story. Some months ago, years ago, I actually did give an example of what happens if you add vinegar to bacteria. So you have to search a little bit in this channel to find this. And of course, vinegar, or the acid rather itself, is able to, of course, also have a disinfecting power. Yeah, I started off with a $30 microscope. Okay, now I have 3 compound microscopes. Looking down at the lens is like entering a new world. Very fascinating. Yes, that's also one of the reasons why I like it. I like to explore things, and sometimes you simply don't know what to expect. So, for example, when I collected those water samples, I was rather surprised to find so many flatworms in the sample. Yeah, so it's something that I did not find before. Yeah. Could you find aphids outside and expose them to capsaicin? Yeah, as I already mentioned, right now we don't have so many aphids, but we used to have quite a lot of them. Okay. If capsaicin react only with receptors in our mucus, does this mean that it has no effect on the stomach and intestine? Actually, I think it does. I mean, in the mouth, we're able to detect it. But what I heard is that some people who are not used to it actually got pretty sick because they got stomach cramps and so on. So apparently the substance does also have, is able to trigger responses inside our digestive system as well. But of course, those receptors are primarily in our mouth because this is where the food is. That's why those receptors are important to have them in our mouth. But it is actually something that I read that actually people, some of them who were not used to it, actually got pretty ill as well because they had got cramps. So evidently also the stomach is able to respond to it. So this experiment made me wonder if these organisms are able to sense heat at all. If they can't, can it suggest that our receptors either evolved separately or changed a lot during evolution? That's an interesting question. I think there's several, without getting too philosophical, there is this thing as follows. Let me try to give a good answer. The question is if you let me give you an example here. Let's say that there is a worm, like I just showed you, in a pond, and the temperature difference of the pond is, let's say, 4 degrees centigrade to, let's say, I don't know, let's say 20 degrees, right? But the pond will never get much hotter than that. So the question is there even a need for the worm to sense temperatures higher than that if this is not part of the natural environment? That's a little bit the question. And this is basically how it ties in with the evolution question, right? So is it possible that organisms are able to sense temperature? I think if it is necessary for their survival, yes, but it does not tell us anything about their subjective perception, right? So it does not mean that it kind of, in quotation marks, feels the same as when we are able to sense temperature. Yeah? Just because organisms are able to respond to something does not necessarily mean that they also have a subjective perception of it. And that's another thing that I just want to mention, right? There are many ways how organisms are able to respond, but they might not be consciously aware of it, and they just respond, right? Because conscious awareness requires a brain and a nervous system and so on. Yeah? And some of these organisms have very simple nervous systems. So a question whether they're even actually able to sense pain in the way that we as humans understand it. It's getting a little philosophical. Can you put a small electric current on them next time? As a matter of fact, I did buy a power supply some time ago. And I did try to expose a flatworm a couple of months ago to the power supply. It didn't like it. So indeed, it is like this that because they have a nervous system, as soon as you switch on the power supply, they will actually make a little twitch and they will respond because the muscles and the nerves, of course, being electrical in nature, they will respond to the electricity. This is indeed possible that there is a response here. According to ChatGPT, most microorganisms don't have the receptor, the pain receptor, so no reaction, only salt and other ingredients. Yeah, salt is generally a big one because it directly impacts osmotic balance and the flow of water, and that is very important. Yeah. So, some insects may have the sensor, maybe some larvae, maybe, but again, the question is whether certain receptors are relevant for their survival, right? That's a little bit, for example, in the case of heat, for us humans, it's important because we're eating hot food and so on. So we need to be able to detect it, obviously, because it's part of our natural environment. But for many organisms, it's not relevant because it's not part of their natural environment. Tardigrades don't have it either, so no reactions are to be expected. What are some cool things one can look at under the microscope? Depends a lot on your personal interest. I like right now I'm doing a lot of microorganisms, microanimals, but what I also recommend to some people is if you buy yourself a set of ready-made slides of plant cross sections, you can actually look at the nice patterns of nature there. Because the plant cells look very regular, then you can actually make posters and hang them up on your wall. That's actually what I've done. So it's going a little bit more into the artistic direction as well. If microorganisms and microanimals, basically, if you've already seen enough of them. We could or should try one day an aphid. Okay, yeah, that's... we have to wait again a little bit when they start to appear, maybe in springtime. You should make a rule that questions or comments at Microbehunter should be related to the current observation and topic. Okay, well, that's has been a request that basically in the past I said it's fine if you also ask off-topic questions as long as they're microscopy related, simply to motivate people to participate in the chat. So this was kind of a little bit my suggestion, but I understand the other one as well here. Yeah, comments at Oliver should be for printing out at last, because one of the main reasons for live streaming and one of the main difference between live stream and recorded video is that there is a continuous two-way communication between you and us. I think this is an interesting point. Thank you for the comment. The reason why I have separated it, and I have not done this all the time, the reason why I have separated this is because there have some viewers requested it to be separated, because I always kept on interrupting myself answering questions. And indeed, I think this is a good recommendation that there should be maybe two different types of questions, maybe questions that are a little bit off topic that I can talk about later, and then questions that I can directly address. Because sometimes, indeed, I've also experienced this with this new format. Sometimes there are questions that essentially make only a lot of sense when I actually show you the things under the microscope. So kind of going back in time is a little bit difficult. So I say thank you for this comment. I'm going to think about this, and maybe this is going to be something that I'm going to do. Yeah, because indeed it's also for me a little bit easier to have a little bit something like a more natural communication. I read a little bit and basically I respond to it. Yeah, and then maybe you have a second part of the video where I talk about off-topic questions. I think that's a good idea. Okay. Do you think it could be related to salt content? Yes, it's quite well possible. Not necessarily only salt, but it could, of course, be also other substances in the chip that could trigger a certain response. You could also make a rule to make a hand emoji for observation-related comments. Okay. Yeah, it's also possible. For me, a little bit, I will just tell you a little bit what I'm able to see here. I'm using the program OBS Studio, and I see the chat on the side. But because there is, of course, only a limited space on the screen, I have to scroll up and down a little bit, and then sometimes I don't know where I left off with answering questions. So that makes it a little bit difficult for me. That's why it might take a little bit of time. And for example, right now, what I have to do is I have to find the last comment here. Okay, so basically with the hand emoji. And I have to find this comment because I would like to answer the questions that followed afterwards, if there were any questions. Okay. Let me have a look here. See? Yeah, there are quite a few. Okay. Yeah, so I've got the hand emoji, okay. And then Alexandre asked me another question about maybe try fresh crushed mint to avoid other ingredients such as alcohol or camphor to test the cold feeling. Ah, that's interesting. Yeah, because some substances have this cool feeling. What was the most interesting or uncommon thing you observed under the microscope? That's a good one. Difficult, because there are so many things that are kind of keep on reappearing. I'll think about that. Generally, what kind of I found sometimes impressive is if certain microorganisms start to hunt other microorganisms. So I've actually, seen certain ciliates that were actually swallowing up other cells, which were sometimes even larger than themselves. And if you see that, or I've seen once a flatworm actually going around really hunting and chasing down other organisms. And that's actually a nice one. Indeed, I already mentioned it before, white blood cells in time lapse. I think that was really fascinating. Yeah? Because you can actually see how they're moving around, how they're following other bacteria. You have to be really patient, and you need a camera because you need to film this for a long time, and then you have to speed it up to be able to see the movement properly. But I found that quite fascinating. So. Some comments. I wish you make more publicly towards your awesome website and forum. We need to build the amateur microscopy community stronger. Yes, that's. I've seen over the past couple of months. I've got a core audience here of around 70, 75 viewers every week, and then usually around 1,000 people more watch the live stream in the following week or two. But it never really goes much beyond that. Yeah. So, On the green screen, try putting the light towards the front wall of the tour setup and let the reflected light lit your face, and then try making background transparent. For me, that works. Okay. Thank you for the comment. I have actually done this before as well. As a matter of fact, for example, what I can try to do is the following. Just a second. Let me see. Yeah, so this is actually now the situation. I don't know. Give me some feedback. Do you like this more? The transparent. You see a little bit more maybe of the of the microscope and of the table. But if you prefer this, then please tell me, and then I can leave it like that as well. Yeah. But sometimes I've seen some cases where I had some problems where also my face got a little bit transparent, and for this reason I just wanted to play it safe. Yeah. But if you prefer this, then I can leave it like this as well. Yeah, actually, it does have an advantage that I'm able to see a little bit more of the table. Yeah? Maybe there are a few more. Side illumination. From Alexander. You mean oblique illumination? Oh, that is okay. This is something that I actually do know. If this is a thing, yeah. Because oblique, if this is the same what we mean, this is basically when the light of the microscope strikes the specimen from one angle, and therefore you get a slightly a 3D effect. It's a little bit similar to DIC microscopy, not quite the same, but very similar. Yeah? And easy to realize, but you have to be patient again, adjusting everything. Yeah, I'm glad you catch you on live. Okay, thank you very much. And I think there are not so many more left for me. Okay, I'm just scrolling down. Oliver, I can't find any organisms other than Rotifers and Vorticella in the area I've searched. Pond, tree, moss, etc. Do the following. Be patient. Really, for example, what I've done here, those water samples that are collected, I've collected from the same pond, but simply from different areas. I generally suggest that you keep on looking, and if that's what you find, then that's what you find. Yeah? That's simply then the environment that you have, and sometimes also over the different seasons, then also the microorganisms change. And also, if you keep the water sample standing inside your room, you have got different light conditions, different temperature conditions, then you also see a progression of different organisms appearing and disappearing. Yeah? So this is something that that's kind of a little bit part of the hobby, trying to find new organisms as well. Yeah, it's better. We see more. Okay, fine. Okay, um, then I've had all possible combinations already transparent like this. Then my videos I have myself in a little circle, and this here as well. Yeah, I tried to find magnetotactic bacteria in 2 different local pond water. I didn't find them yet, but got nice a surprise, new stuff found. Magnetotactic bacteria I also tried to find. I also was not successful. And what you do is you place some a water sample, basically with some pond sediment as well in a jar, and then on the outside you place a strong magnet, a very strong magnet, neodymium magnet, and then if magnetotactic bacteria are present, then you should actually see them accumulate on the inside of the jar because they will move towards the magnet. Yeah, so this is kind of a little bit the idea. I have also tried it, also was not yet successful, but I've heard that this should be possible. And ladies and gentlemen, I think I'm going to call it quits again for today, unless you have some more questions. Okay. Yeah, exactly what I was doing, but I'm still trying. Yes. Yeah. So at least in my sample, I was not able to find a magnetotactic bacteria, but I've seen a YouTube video by another microscopist who basically was very successful. Yeah. So I think I'm going to call it quits for today. I hope, and I don't know if I will be successful, I hope that I'm able to show you next week. I don't even know where I put them here, that the diatom extraction technique with hydrogen peroxide. This is still something I'm working on. The sample in here is now white, completely bleached, but originally was dark green because there were algae with diatoms in here. But I think not everything is yet finished. Yeah, and I think I need to get myself a stronger hydrogen peroxide solution, and then maybe it's going to work a little bit faster and a little bit better. But that's essentially one of the things that I still have on the program, and which I would like to show you sometime in the upcoming live streams. And for right now, I'm really not going to call it a day. I wish you nice remaining Saturday and Sunday, and all the best. Happy microbe hunting. See you around next time, and bye bye.